Hi friends, welcome to Science Jagat. Today we are going to talk about CRISPR-CAS. What is CRISPR-Cas system, mechanism of action, types of CRISPR-Cas system, general protocol, and advantages and disadvantages of CRISPR-Cas system. So let's see what it is. So CRISPR-Cas system is a prokaryotic immune system that confers resistance to foreign genetic element, such as those present within plasmid and phage that provides a form of acquired immunity. So CRISPR stands for clustered, regularly interspaced, short palindromic repeat. These are short pieces of specific DNA. In between of these pieces, there are pieces of DNA of viruses that were acquired during previous exposure of the viruses. And what is CAS? CAS is CRISPR associated protein. CAS protein specifically recognizes CRISPR and it helps in recognition and degradation of the target DNA. Okay, so let's see what it is in detail. So CRISPR is a family of DNA sequence in bacteria and archaea, and the pieces contain snippets of viral DNA that have attacked the bacterium earlier and show adaptive immune response. So you can see here CRISPR, it is a segment of DNA in which there are present a stolen phage DNA, which is due to the previous attack of red phage. So it is a palindromic repeat sequence. You can see here it is a palindromically repeated. Each sequence is followed by a short spacer DNA segment. These are the repeated short spacer DNA segments of foreign DNA from previous exposure. Okay. Just next to it, a small cluster of CAS gene is also present. So this whole is a bacterial cell. Now let's see how bacterial adaptive immunity works. So it is divided into three stages, and this is the mechanism of action of CRISPR. So stage first is known as acquisition or spacer acquisition. Here, insertion of a short sequence of the invading DNA as a spacer sequence into the CRISPR array goes on. After this, it comes to the second stage where cRNA processing happens. How does it happen? What is cRNA? So there are three components of CRISPR. First is protospacer adjacent motif. We call it as PAM. CRISPR RNA. It is cRNA as it is written here, cRNA, CRISPR RNA. Third one is trans-activating cRNA. It is cRNA. Okay, so we were on the stage second. Transcription of precursors cRNA. We also call it as pre-cRNA that undergoes maturation to generate individual cRNA. Each of them is composed of a repeat portion and an invader target spacer portion. Okay, now the stage third, we call it as interference. So cRNA directed cleavage of foreign nucleic acid by CAS protein at sites complementary to the cRNA spacer sequence. So here's the structure. See, the CAS operon consists of CAS1, CAS2, CSN2. These three helps in adaptation, or we can say that acquisition, and Cas9 and tra-cRNA helps in maturation interference. And here is the spacer array of CRISPR repeat. Okay, so this is the Cas defense mechanism. Let's see. As I told you, there are three stages. So let's simplify these stages. First is adaptation. Here, the insertion in this stage, it leads to insertion of new spacer from the foreign genome in the CRISPR locus. As you can see here, the phage genome, a part of phage genome is inserted in the CRISPR locus. Then the second stage goes on, that is expression. In this stage, transcription and maturation of that cRNA happens. Okay, so in the second stage, expression of Cas gene and transcribing the CRISPR into pre-cRNA. So this is the transcription process, and this all happens with the help of Cas genes. So the pre-cRNA is processed into mature cRNA, by Cas protein and necessary factors. Then comes the third stage, interference. Here, in the last stage, target nucleic acid, the target nucleic acid is recognized and destroyed. Okay, so Cas protein CRRNA complex binds and degrades the invaded DNA. See, so we have seen the simplified 3 stages. In these 3 stages, there are few CRISPR-Cas systems. So there are 3 types of CRISPR-Cas system which works. So let's see how does it work. As you are seeing here, this is the phage infection. The first stage, spacer acquisition. Okay, in this spacer acquisition, there are 2 Cas which helps in this process. Cas1, it is a nuclease and an integrase, and Cas2, it is an endoribonuclease. Okay, now comes the second stage where cRNA transcription or cRNA biogenesis and processing takes on. Okay, so in this process happens in 3 forms. So that is type 1, type 2 and type 3. You can see here there are type 1, type 2 and type 3. So what are the difference in these systems? In all the process, in all the 3 types, cRNA biogenesis and processing would happen, but the difference lies within the protein helping in the process. Okay, so in type 1, Cas6E and Cas6F is working. In type 2, RNase 3, and type 3, Cas6 and homolog. These systems are working. Okay, now in the next step, target degradation. In type 1, PAM and cascade complex is helping in the recognition and degradation of the target genome. In type 2, PAM and Cas9 is helping, and in third, type 3, CSM complex and CMR complex is helping in the recognition and degradation of the DNA and RNA from the target. Okay, after this process, the end repairing or the DNA repair by 2 processes. First is non-homologous end joining and another one is homologous rejoining. Okay, so these are discussed in the previous video of gene editing, and I have also discussed the process of ZFN. So you can go there and watch the videos to understand the mechanism. Let's move forward. Okay, so this is the structure of Cas9 complex and it is inactive right now. And this is the structure of cRNA. So this is target complementary region. This is loop 1. This is loop 2, loop 3 and loop 4. Okay, both of them meet together and form an active complex which help in recognition and degradation of the target genome. Okay, so this is called as guide RNA and this is target complementary region. Okay, so let's move forward. Now here is the general protocol of CRISPR. Okay, so what is the protocol of doing CRISPR in the laboratory? So first of all, we will select a genome target. It should be 20 base pair sequence which is followed by PAM. We can use online tools to minimize off targeting. And second step is designing of sgRNA. So sgRNA is expressed using a small RNA promoter such as U6P or U3P. Here you can see this diagram. It's target sequence which is followed by PAM. It is 20 base pairs. And here is sgRNA backbone with the guide sequence U6. Okay. Then next step is first nucleotide in the guide sequence is a G guanine if U6P is used or adenine if U3P is used. So in case of U6P it should be G and in case of U3P it should be A. Okay, so guide sequence should match the target except for the first nucleotide that does not have to match. Okay, now the third step is assembly Cas9 or sgRNA construct. So here assembly. You can see the assembly of sgRNA and Cas9 in a single stretch. Fourth is delivery of two plants. So the constructed genome would be transferred to the plant. This could be done by protoplast transformation, agrobacterium transformation, callus bombardment, and there are some few more methods to do this. Now, the fifth one is regenerate and screening transgenic plants for gene editing events. So first of all, the delivery of plants, and to check if the delivery of plants is successful or not, we do screening for mutants. Okay, so first is restriction enzyme site loss, second one is survey or assay, and the third one is next generation sequencing. So by these three processes we can check or we can screen for the mutation process. Okay, so there is a comparison between prokaryotic and eukaryotic system. We have just discussed the prokaryotic system in detail. Like you can see here, in transcription process, in the processing of cRNA transcript, RNase 3 is used, Cas9, and an unidentified nucleus is used, which is a necessary factor, and the DNA degradation is going on with the help of them. Okay, but here in eukaryotic system, engineered CRISPR-Cas transcription is with the help of sgRNA, which is engineered here. Okay, and then this sgRNA will help in nuclear and with complex with Cas9, and the complex will help in the cleavage and the recognition of the target locus. So the difference lies in sgRNA and the other type complexes, the type 1, type 2 and type 3 for prokaryotes. Okay. So there are different Cas proteins and their functions. I'm not reading all these functions, but you can pause the video and check all these proteins and their functions to differentiate between all of them. Cas1, Cas2, they are universal space acquisitions. Cas3 is for target interference, DNA helicase, and it functions as a DNA helicase and endonuclease. And type 4, type 5, type 6, type 7 work for different types of CRISPR-Cas system and expression. So you can read it whenever you get time. You can pause the video and read it. Cas9 is type 2 signature, and it can work on multi-domain protein with MRC, H and H nucleus domain. And Cas10 is type 3 signature, CRRNA expression and interference. And here are some examples of crop modified with CRISPR technology. It's corn, rice, sorghum, sweet orange, tobacco, and wheat, and potato, soybean. Okay, so there are some advantages. This technique is actually simple, accessible, cheap, and more versatile than ZFNs and talons. It creates genetic variants for a desired locus. CRISPR-Cas9 also allows multiple gene editing by the simultaneous expression of 2 or more sgRNAs. It allows the inexpensive assembly of large gRNA libraries so that the CRISPR-Cas9 system can be used for high throughput and functional genomic applications, bringing genome editing within the budget of any molecular laboratory. CRISPR-Cas9 system can introduce biallelic or homozygous mutation directly in the first generation. So being an easy and affordable tool, CRISPR-Cas9 promises to revolutionize basics and applied plant research. There are some drawbacks also. So one of the few drawback is that the CRISPR-Cas9 technology is the relatively high frequency of target mutation reported in some of the earlier studies, and the limited data available thus far suggests that off-target effects are rare in plants, which can be eliminated by backcrossing. So off-target effects vary even among different cell types in the same species. Okay, in the next video I am going to discuss about talons. So please subscribe the video and like and share if you like the video, and you can go and watch gene editing video and some videos on biosafety also. Thank you so much for watching the video. Bye.